Okay, it's just past 2 o'clock and people seem to have mostly come in. So we're going to start. So this is the Genomics England Research Seminar Series. I'm Tim Hubbard from King's College London and Genomics England, and I'll be chairing this seminar today. Next slide, please. So we've got 2 speakers, Professor John McGrath, also from King's College, talking about the skin GECIP, and we've got Dr Marta Futema from George's University talking about FH. So we're going to start with John. Just going to say before we start, this is going to be recorded. If you want to ask a question, you can put your hand up, but we'd prefer you to type it into the Q&A, and at the end of the talk, I will direct those to the speakers. And if we don't have enough time because there's too many questions, then we'll try and get those answered in the Q&A as well, electronically. Just to advertise a bit the next research seminar, this is going to be in June. And again, we've got 2 speakers talking about patients and their families, Sakia and Celine, and then Nick Owen talking about some novel human ocular genes through evolutionary analysis. So that's a bioinformatics type of talk. You can keep up to date on all of this by following the Twitter. There's obviously the main Genomics England Twitter, but there's also a specific one for the GECIP, the Genomics England Clinical Interpretation Partnership, where all these research talks are from members of that consortium. So I will now hand over to John McGrath, who's going to give the first talk for about 20 minutes, and then we'll have questions about that talk. So over to you, John.
Thank you very much, Tim, and good afternoon, everybody. Yes, John McGrath. I'm from Guy's Hospital in the tower block on the 9th floor, sitting almost on top of Tim's head, in fact, in terms of where I am today. This is a relatively short presentation just on behalf of one project within the skin GECIP, and you can see a number of individuals who were involved with this, and I'd like to acknowledge all of those and also everybody at Genomics England who's helped with this particular project. So here is a starter. Here are the genodermatoses, the inherited skin diseases, those Mendelian disorders, and there are about 1,000 different Mendelian disorders that affect the skin. Now, within the 100,000 Genome Project, we were allocated about 1% of those genomes, which gave us a good opportunity to explore certain mysterious genodermatoses in the field of diagnosis and discovery. Just before I kick into this individual project that I'm going to describe, just a very brief history of time when it comes to skin genetics and discovery, because we as dermatologists have that distinct advantage of being able to view and sample the organ that we're interested in. So for many years, people like me have followed a candidate gene approach where we can look at the skin. And so for cases like this, where there is blistering and hair and nail abnormalities, we can actually look at skin and search for candidates. And for many years, this was our preferred modus operandi. So we could take a skin biopsy. And here you see a picture of this patient's skin. And the key abnormality is in the epidermis, that brick wall-like structure where the cells are starting to fall apart, the gaps, the white gaps between the cells. So we can go from light microscopy into electron microscopy and show that the zippers, the junctions between the cells, called the desmosomes, are not functioning properly. And then we can go to immunohistochemistry to search for our candidate to see whether there's a desmosomal gene that's aberrantly expressed. And this approach has been incredibly useful for recessive diseases where we think there'll be loss of function, loss of expression of the encoded protein. So in this case, from clinic to biopsy to ultrastructure to immunohistochemistry, we come up with a desmosomal protein called plakophilin-1. We then conventionally sequence, find the nonsense mutations accounting for the loss of expression, and we make up a name. In this case, ectodermal dysplasia skin fragility syndrome, the first disorder of desmosome cell junctions. Before Genomics England and next-gen sequencing, our other option for autosomal dominant pedigrees, particularly large ones, was to go for genetic linkage. So taking pedigrees like this, where you see telangiectasia, dilated blood vessels, and the picture of the mouth is showing a soft palate squamous cell carcinoma. So large pedigrees, where we do microsatellite markers, we look to see which bits of the genome are shared by affected individuals. We then do fine mapping, in this case down onto part of chromosome 3, sequence a fair number of genes, identify an abnormality, and we have our gene. And we can then come up with a diagnosis, in this case a new condition, which we chose to call telangiectasia oropharyngeal cancer syndrome. So that was how we used to do it in the old days. And the advantage of skin being that we can actually access it and look at it very readily. Of course, with the era of next generation sequencing, it's all change in dermatology, just as it has been for every other discipline as well. And so very briefly, just to recap what we've done in dermatology, is that over the last decade or so, there have been 166 new skin disease gene associations matched up. And a number of these were just old diseases matched up to new gene discoveries. But there were also about 35 brand new genodermatoses, so about 3 per year of brand new conditions. Here, for example, is one of those brand new conditions. This is an autosomal recessive inflammatory skin condition associated with loss of function mutations in epidermal growth factor receptor. You can even see the pustular rash on the right-hand side, and this is very reminiscent of EGFR inhibitor drug rashes that patients in oncology receive. But this is the germline abnormality in epidermal growth factor receptor. And here's one more example of loss of function mutations in a different desmosomal protein called desmoglein 3. Dermatologists know desmoglein 3 as a target antigen in the autoimmune blistering disease pemphigus vulgaris. Well, here is the genetic equivalent for this autosomal recessive loss of expression, with the oral and mucosal and upper airways erosions and some hair abnormalities as well. So these sort of matchups over the last decade or so, and the breakdown of these skin diseases look like this. It's about half dominant, it's about half recessive, and of those dominants it's about half de novo dominant, and the other half have vertical transmission, just a small number of X-linked disorders. And within the spectrum of diseases, the wagon wheel shows it's pretty much every type of genodermatosis that's been characterized. The big quadrant in gray here are developmental disorders. And I guess in clinical genetics, that's been the problem for most of us over the years, trying to sort out those weird developmental disorders, of course, helped by next-gen sequencing. In terms of progress, starting back in 2009, you can see that there has been considerable progress. It goes up and down a bit. Maybe it peaked a few years ago, but still new genodermatoses are discovered. Of note, because most things are done in private research settings, it's been WES, whole exome sequencing, rather than whole genome sequencing, that's heralded the major success in discovery. And as a pipeline, we can pick our favorites, but every year there's something new and exciting that appears in genodermatology. There have also been several cases where we've got multiple mutations affecting phenotype. Sometimes it's double genodermatoses, and other times it's genodermatoses mixed in with some other genetic disease. Let me just illustrate a couple of those for you. So epidermolysis bullosa, as most of you will know, is a collection of inherited blistering diseases that's divided into many different subtypes. But here's an individual from a consanguineous family who happens to have double EB. Simplex and junctional, two different types of EB, autosomal recessive versions thereof. Very unfortunate for this individual, but that's the power of this reductionist approach of sequencing to sort out these double diagnostics. Here's another double diagnostic. There's a developmental abnormality of hair and a developmental abnormality of nails. And dermatologists are pre-programmed to think that must be a form of ectodermal dysplasia, where we combine those ectodermal anomalies. But actually the reductionist approach to next-gen sequencing shows it's two separate autosomal recessive disorders in this consanguineous individual. PADI3 for the hair and R-spondin for the anonychia, the nail abnormality. And just one more of skin and something else. At the top is something that dermatologists will recognize as a form of cutis laxa, with that wrinkly palm and the arthrogryposis, the thin-looking skin with the prominent veins, the large low-set ears, but this patient has proteinuria, nephrotic syndrome. It's not a new version of cutis laxa, it's two separate autosomal recessive diseases sorted out thanks to next-gen sequencing. And there are even triple diagnostics where you can take the reductionist approach to an extreme and really dissect the phenotype and come up with a specific genetic abnormality. So lots of progress in the research labs, and of course the Genomics England 100K project has also contributed to this decade of discovery. And so the clinical impact, of course, of next generation diagnostics is very much around better treatment for patients and also better care in the clinics. And so I just illustrate one example because this is relevant to the case history I'm going to talk about today. This is a Middle Eastern child with inherited poikiloderma, so dyspigmentation and some dilated vessels. And we have a differential diagnosis as pediatric dermatologists for this type of condition. And here you can see those conditions listed here. But what do we do in clinical practice in the old days? We watch and we wait. And when we watch and we wait, we see various abnormalities in terms of growth and hair and skin and mouth and palms and soles and so on. And we're just left waiting for a diagnosis. We're trying to think with the passage of time, what will it be? So we switch to next-gen sequencing and we find in this case a de novo heterozygous mutation in FAM111B. And that gives us a diagnosis based on the literature of this complicated term described by French colleagues: hereditary fibrosing poikiloderma with tendon contracture myopathy and pulmonary fibrosis. And here is the original publication from our French colleagues showing in white Caucasian skin this poikiloderma and the sclerosing changes in muscle and tendon and so on. So of course now we can become, with this genetic information, better clinicians, because rather than just looking and photographing, all of these features here are part of this particular syndrome. Moreover, everything that this child has in yellow is what they have now. But this is a condition that takes off during puberty. And so everything in white I can specifically look for, so I improve my clinical acumen by monitoring this particular condition in this way. And I also know that pulmonary fibrosis will be a life-shortening complication. And so I have a window of opportunity to do something for this patient. And that do something is to think about mechanistic-based treatments. In this case, it's about using metformin and aspirin to try and help the individual. So that next generation sequencing information has been so powerful in clinical management, both in the clinic and what we can prescribe for patients like this. Which brings me to today's story, this brand new autosomal recessive genodermatosis, which we're choosing to give a new acronym, LIFAC syndrome. So as you'll see, hopefully, this stands for LTV1 associated inflammatory poikiloderma with hair abnormalities and acral keratoses. Let's put some flesh on that. Well, these are the two UK pedigrees that have been studied, a British Pakistani family and a British Indian family, not known to be related to one another, but both from the Midlands area. So the Pakistani family known to be consanguineous with 3 affected individuals. The Indian family not known to be consanguineous with just the 1 affected individual. Let me show you the clinical features. So I'm illustrating here one individual from both families at different ages, so you can match up the colors and the years and see what's going on. But you can see the shared common clinical features. There is a poikiloderma. So by poikiloderma we mean hyperpigmentation, hypopigmentation, telangiectasia, and atrophy. That's the dermatology definition of poikiloderma. So you can see that in these pictures at the different ages. But there are other features too. Scalp hair and eyelashes and eyebrows sparse or absent. There were some punctate keratoses, so little horny lumps on the palms and soles. There were also some ophthalmological abnormalities in some of the individuals, and the skin biopsy just confirmed that there was indeed a poikiloderma. So back to our differential diagnosis. This is the patient's own palms this time, and none of these particular entities seem to fit. So these individuals were recruited separately to the 100,000 Genome Project by various colleagues. And various panels were applied. So patients 1, 2, and 3 from the Pakistani family had those panels applied, and different panels for the Indian family patient number 4. But by scrutinizing those panels, unfortunately, there were no pathogenic variants, and so the next stage was a gene agnostic approach. And when that was done, this variant popped up in a gene called LTV1. This stands for low temperature viability protein 1, and it was present in affected individuals in both families. More than that, patients in the Pakistani family were all homozygous. Parents were heterozygous, and the same homozygous variant was also present in the Indian family as well. And with the advantage of whole genome sequencing, of course, we're able to look for a common ancestral allele and identify a block of homozygosity consistent with that idea. This is not a gene that I have heard of before. So what do we know about low temperature viability protein 1 and this particular missense variant? What and how are what we as clinicians are always going to say when it comes to a genetic variant in explaining the phenotype. When we look at what's in the literature in terms of knockout mouse, lethal before weaning. If we look in zebrafish, also embryonic lethal, but maybe something to do with gut and blood. When we look in yeast, maybe some ideas. It seems to disrupt the ribosomal complex, upsetting the translational fidelity of a number of mRNAs on their journey to becoming proteins. So I guess we conclude from the literature that it's probably got something to do with ribosomes. Now that's not that straightforward, is it really? Just a quick refresher about ribosomes. You probably remember all of this in terms of the journey from mRNA to protein. But in terms of the machinery, it's complex. There are more than 100 different ribosomal proteins and more than 200 support proteins underpinning this complex structure. So the dynamics are complex. This is just a snap sketch of part of that ribosomal machinery. What we know is that LTV1 is somehow involved in the small ribosomal subunit assembly. But in terms of disease and clinical genetics, there are no previous human genetic disease. So in terms of this variant, if we go back to genetic analysis and think what we can find, if we look on NOMAD, there is just one heterozygote in about 250,000 alleles, no homozygotes, and that includes, of course, 30,000 South Asian individuals. When we use the in silico tools like SIFT or polyphen2, it predicts that this missense change may be benign. But when we apply splice AI and maxent scan, it predicts that this single nucleotide change might actually create a cryptic new donor splice site. And so we looked at that. So firstly, in mini-gene studies, you can see that there is indeed an abnormality. Let me take you through this. So in wild type, that RT-PCR product is of that particular size. In the next column is the patient, and there's very little normal sized PCR band. Instead, there is a truncated, smaller band. And if you move to the lower picture, bottom left, you can see in red where the point mutation is, and it creates this cryptic donor splice site. So the end of exon 5 is lost, and this is out of frame. We can then go back to the human skin and show the same cryptic out of frame splicing, here with semi-quantitative RT-PCR and the predicted stop codon. So this particular missense variant is much more than that. There is reduced gene expression, LTV1 expression. So this is the working hypothesis that somehow this small subunit of the ribosome is messed up. But of course, we're still wondering why skin and why poikiloderma. So what clues can we achieve? Well, if we look at the expression of LTV1, it's pretty much everywhere. More than 200 tissues express this particular protein. But you can see in blue, skin is quite prominent in terms of its expression compared to other organs. The next question, of course, is where is it expressed in skin? And this is where a surprise and perhaps some reassurance for dermatologists comes in. So I'm showing you here immunofluorescence with a commercially available antibody. The top two panels are control skin, and the bottom two panels are patient skin. And if you look in the control skin, top left to start with, you can see this diffuse cytoplasmic, maybe nuclear, and also some punctate staining around the nucleus. And then top right, you can see that there's continuous staining in the basal layer of the epidermis, right the way across the section. In the bottom left, you can see that the intensity of the staining in patient skin is just slightly reduced. But in bottom right, perhaps more convincingly, you can see it's somewhat patchy in terms of the continuity of that basal keratinocyte layer staining. So we've got some clues that a protein is in the right place to explain a poikilodermatous abnormality. And we get some clues perhaps to function from these predicted interactions in terms of the type of repair processes and cell cycling processes and the cell dynamics that might explain the phenotype. Actin cytoskeleton, very relevant to poikilodermatous disorders. So bringing the story to this sort of conclusion, we seem to have a ribosomopathy. Now, ribosomopathies are a recognized group of conditions. They're very heterogeneous, and some of those other proteins have been implicated in those. Usually, as a general term, the mutations in ribosomopathies give rise to haploinsufficiency, and they mess up either specific mRNAs or the function across various different types of ribosomal protein processing. Now, we don't normally see ribosomopathies in dermatology. It's normally to do with craniofacial skeleton, hematopoiesis, digestive organs. It's not really a skin problem, although we do have some link in the hematopoiesis sector. For example, dyskeratosis congenita is one of a condition that we're familiar with as dermatologists, and DKC1, one of the old genes for dyskeratosis congenita, is indeed a ribosomopathy. And one of the newer genes implicated in dyskeratosis congenita is also linked to ribosome pathology. So we have a story there that's emerging. Clearly still a lot to do, but this is the new syndrome that is now appearing. Lifeact syndrome, a new autosomal recessive ribosomopathy. Of course, whilst we're sorting this out, and indeed this one is in fact IMPRESS in human molecular genetics, we continue to work through the 100,000 Genome Project and other conditions, and working with colleagues in Birmingham, Manchester and London. This is a condition that we're working on at the moment, skin fragility and woolly hair, and hopefully we'll have the answer to that soon. But in the meantime, I thank my various colleagues who told me a lot about LTV1 and are involved in the ongoing work, and I thank everybody involved at Genomics England and the 100K project for helping us bring this particular story to fruition. And thank you very much for listening to this story.
Thanks very much, John. So people got questions, you can put them in the Q&A, or you can put your hand up. So I'll start with one question. Generically, this was a nice story of where the panels didn't give the diagnosis, and you had to do this extra deep diving, looking at all the genes across, and of course you had multiple patients. What fraction of the skin conditions in the 100K project have actually got a diagnosis? How far away are you from 100%?
Yes, that's obviously a good question, because we'd like to know whether it's a research tool that we're doing in terms of our own privately funded research and whole exome sequencing, or whether we're using part of these national initiatives as well to crack the cases. I feel that the predominant tool is whole exome sequencing in private research projects, just because we had ongoing support from Wellcome Trust and other institutions, and we kind of got used to doing that. Of course, we picked the low-hanging fruit perhaps with some of those cases, and I'm sure we can turn to whole genome sequencing and some other types of sequencing to solve those remaining mystery cases. But across those of us in dermatology genetic research, probably 90 to 95% remain whole exome sequencing in research-based projects, and there are just a few cases cracked by 100,000 Genome Project. Perhaps if we'd been given more than 1,000 genomes, 1% of the project, for a condition that affects about 20% of phenotypes, we might have turned to it a little bit more. But there's still work in progress, and I think this the woolly hair skin fragility one that we just highlighted is going to be yet another story. So a few, Tim, is the answer in terms of success so far.
A few are undiagnosed, or a few are diagnosed.
A few have been diagnosed,
Right,
but of those, all those matchups that
of
I
the
told you,
1,000.
yes, of those 166 cases that have been solved in the last decade or so, the vast majority, 90%, are whole exome sequencing, and most of those will be through private research projects, not projects such as 100,000 Genome Project.
Right. So, given that everybody inside the 100K is a clinical patient, just directly recruited from the NHS, a large fraction of those are still undiagnosed.
Exactly. So there is definitely a need to continue to work on those cases and hopefully crack them, because we should be able to offer diagnoses to our patients, and we still are missing quite a few.
Okay, so a few questions. How strong is the connection to ribosome biogenesis? Is there any evidence beside the study in yeast, any known protein domains in LTV1?
Yep, no, we don't have any particular strong biological link other than what I've shown you in this so far. So it's work in progress, and hopefully we'll carry on with that cell biology and try and link to some real story and understanding of domains, function, and dysfunction.
And is LTV1 druggable?
Not at the moment, as far as we understand, but again, that's something that we are looking to do. Repurposing of drugs is very big for us in dermatology at the moment, and hopefully we'll be able to influence some of that story, but that's also on the agenda. And
there's a comment, nice to see ribosome finally having an identifiable fault. Do you think there are any other parts of the inner cell that haven't yet caused a genetic skin disorder that can be looked at?
Absolutely. I mean, we have so many organelles and activities that are going on. There's so much even within the cytoskeleton that we don't really fully understand. We've got some exosome disorders that we're working on as well at the moment, and I'm convinced that we will have other mysteries which will sort out disease, but also give us great new insights into physiology and pathology of keratinocytes, fibroblasts, and other cells in the skin.
There's a comment here which I'll turn into a question. Identifying hereditary skin disorders can be challenging because both the rarity, lack of awareness, and particularly when skin disorders are not identified at birth. Is this potentially a candidate for the newborn screening? I mean, the newborns were going to, basically try and spot things that might have an impact in the future. Some of those, of course, are linked. You want to link it to a test you can carry out. Are there any candidates for that where you could see a mutation that might influence a skin condition and then actually do a test after you can get it in the skin and observe something?
Yes, just breaking up slightly, Tim. So that was about newborn testing and whether there's any value in early diagnosis, whether it might influence clinical course. Is that?
Yeah.
Yes.
Yes.
Yes. Yes, we do have a number of those cases. For my own specialist interest of epidermolysis bullosa and skin blistering, there are many of those conditions that we now can anticipate extracutaneous abnormalities, some of which may be life-threatening. I'll give you one particular example. There's a gene called KLHL24, which is a Kelch-like protein, and that causes relatively minor blistering in children. But there is a big risk because Kelch-like protein 24 is also expressed in the heart, and these individuals are at risk from cardiomyopathy, arrhythmia, and sudden death in teenage years and onwards. So sometimes we discover things in the skin that have even more profound implications for imminent health activity, and that would be one example of those. So we have a number of others where we can line up what's going on in the skin. Maybe it's going to tell us about other organs, including brain and eyes and so on. So yes, it's very much part of our remit to make early diagnoses and then prognosticate and carry out primary or secondary prevention where we can.
Okay, I think probably we have to leave it there and move on to the second talk. John, you can go and look at the Q&A. There's possibly one or two you might reply to in there, but otherwise we've covered everything. Okay, thanks very much, John. Now it's time for Marta, and she's going to talk about FH.
That's right. Thank you for introduction. Can you hear me?
Yes, can hear you fine.
Where do we go?
Yeah. We can. Okay. We're seeing the wrong screen. We're seeing your note. That's better. Yep, working now.
Great. Thank you very much for the introduction. My name is Marta Futema, and I'm here today with Martin Bird, who is my PhD student here at St. George's at the Cardiology Research Center. So today we will be talking to you about FH and what we have found in the 100,000 Genomes Project. So I would like to start by telling you what FH is in very simple terms. Familial hypercholesterolemia has it in its name. Its major characteristic is that it affects families. And this is really one of the most important characteristics of FH. It should not be ignored. FH can be a devastating effect, can have a devastating effect on families if undiagnosed. And it is a disease of high bad cholesterol, a symptom that is often silent. This is due to, not due to bad diet, but to because of a defect in one of the genes that regulate the clearance of cholesterol from blood. So the high cholesterol over time leads to premature heart disease. And treatment to lower cholesterol is available. And if implemented early and correctly, it reduces the risk of heart disease. So that the DNA isn't necessarily your destiny if you have an FH mutation. If you are directly affected by FH or would like to or know someone who is affected and you would like to know more about it, there are two major organizations. In the UK, we have the Heart UK, the cholesterol charity, and in Europe, there is FH Europe, a network of patients who are led by two very passionate women who provide loads of support for families who are affected by FH. And they also have booklets available in several different languages suitable to age group of patients. And of course, the British Heart Foundation has been supporting FH research over the years and also cascade testing of families affected. Lastly, if you are a patient and would like to offer me some feedback, please, on the ongoing work or any future work that you think might be relevant, please get in touch. But today I will talk about our initial findings in the cohort of FH patients who are part of the 100,000 Genomes Project. But before I get to that, let's have a look at more details in terms of FH. So its clinical characteristic, it is an autosomal dominant disease, therefore 50% of the first degree relatives are likely to be affected. This means that FH patients have a strong family history of premature heart disease or hypercholesterolemia. And as mentioned before, FH is a disease of high LDL cholesterol. And so they are exposed to it from birth. Therefore, there is a lifelong exposure to these high levels of LDL, and that leads to significant burden of LDL. So let's have a look at this graph. Here in blue, we plotted the burden of LDL cholesterol in FH patients, and in red are those of a normal population. We know that LDL cholesterol goes up with age. But if you take, for example, the burden of LDL cholesterol in FH patient at the age of 45, you will see that it corresponds to the burden of non-FH 70-year-old person. So it is clear that the LDL cholesterol accumulates in FH much earlier. And that leads to increased risk of premature heart disease, myocardial infarction. And it has been estimated that 50% of men and 30% of women will get CHD by the age of 55. Recent studies that employed sequencing technologies on population scale showed that FH mutations affect 1 in every 250 or 300 individuals. In the UK Biobank, we have a study currently ongoing. We observe frequency of 1 in 280. This frequency of FH mutations. So that makes it the most common Mendelian disease. Yet it remains highly underdiagnosed worldwide. Here is a figure from 2013 showing that only 12% of FH cases in the UK were diagnosed. Now, this figure is based on the old FH frequency, which we used to think that it affects 1 in 500 individuals. So with more data, we know that FH is more common, therefore the situation of underdiagnosis is likely to be much worse. And the current estimates that FH affects 7 to 8%, that we are currently know of 7 to 8% of FH cases in the UK, it's not a great result. So we are missing a great opportunity here to prevent heart disease, since studies have shown that introducing statin treatment from as early as 10 years of age leads to 15% decrease in LDL cholesterol burden by the age of 18 years. This is if the child is treated by statins. Therefore, early diagnoses are crucial for CHD prevention in FH patients. And the NHS long-term plan includes, one of the priorities is to increase the FH diagnosis to 25% over the next 5 years through the NHS Genomics program. So yes, early diagnosis is definitely important. So in clinical setting, FH can be diagnosed using the Simon-Broom criteria. A patient would have a total cholesterol of LDL or LDL cholesterol over the mentioned here thresholds. The thresholds are lower for children. And assessment of family history of high cholesterol or premature heart disease is also an important criteria. So these two would classify a patient as a possible FH. Now, more severe cases will also have phenotypes indicating accumulated cholesterol deposits, such as tendon xanthomas, and presence of those or an early or already identified family mutation classifies the patient as a definite FH. Other clinical criteria include the Dutch lipid clinic scoring system or the US MedPed criteria, but they all seem to be performing equally well or equally bad, depending on the point of view, in identifying FH mutation carriers. Now, overall, 40% of those with possible FH diagnosis using Simon-Broom criteria and 80% of definite FH are found to actually carry an FH mutation in one of the known genes. So the genetic causes of FH are due to defects in genes that encode major proteins involved in the clearance of LDL cholesterol from circulation. And these include the LDL receptor, which is kind of like a seven-fingered hand expressed on the surface of mainly liver cells that binds LDL cholesterol. There's ApoB, which is an apolipoprotein that wraps around the LDL particle, enables its transport in blood, and it acts as a ligand to the LDL receptor. And another important player in this pathway is the PCSK9 that regulates the recycling and degradation of LDL receptors. Therefore, a gain of function mutation in the PCSK9 leads to increased degradation of LDL receptors, resulting in a smaller number of receptors being available to bind LDL cholesterol. Recently, a single mutation in the ApoB gene has been suggested to cause FH, and I will talk a little bit more about that later. It is important to mention that there is a great value in studying genetics of FH, since some findings have significantly contributed to the development of novel drugs, especially the very successful PCSK9 inhibitors. So if we look at the frequency of FH mutations, we find that the majority of mutation-positive cases will have a mutation in the LDL receptor gene. These also include large rearrangements, such as structural variants. However, the most common single FH-causing variant is located in the ApoB, and it accounts for 6 to 10% of all FH mutations. Least common is the gain of function mutation in the PCSK9, which has been associated with the most severe risk of premature CHD. The frequency of the mutation in ApoE is largely unknown, as the findings are still relatively new. However, our work in the UK Biobank shows that it is present in about 1 in every 10,000 individuals, and it accounts for about 2.6% of all FH mutations, at least in the UK Biobank. Overall, there are over 1,700 FH mutations reported in those genes, and you can find them on the mutation database, although more accurate variant interpretations might be found via the ClinGen website. The bottom line really is that the cause of FH is highly heterogeneous in the UK, though there are some populations where the majority of FH cases are due to just few mutations. So although we treat the phenotype, the FH mutation is a crucial piece of information for early diagnosis and cost-effective family screening, so-called cascade testing, which is recommended by the NICE guidelines. Okay, going back to the 100,000 Genomes Project, here we have 544 participants sequenced as part of the rare diseases cardiovascular domain. They include 67 relatives, of whom all are affected. And one of the criteria for inclusion of a patient for whole genome sequencing was that they were supposed to be sequenced for variants in the known FH genes, therefore supposed to be mutation negatives. When we look at the self-reported ethnicity, the majority are white British, but we also have a representation of all other major ethnic groups in the UK. So the first step of the analysis was to look at sequences or changes in the genes that are currently known to cause FH. So we started with the rare variant analysis in the LDL receptor gene. And I think first it was a big surprise that we found 25 different likely pathogenic variants. Furthermore, there were 8 different clear pathogenic variants and 5 novel variants that were predicted either likely pathogenic or pathogenic. So therefore, by analyzing LDL receptor, we were able to explain FH in 51 probants and 7 relatives. All of those variants should have been detected by the standard genetic testing, which is mainly based on targeted sequencing. In addition, we have also identified further 4 variants of unknown significance in that gene. Next, we wanted to see if the whole genome sequencing can detect large structural variants. And we know that large rearrangements in the LDL receptor account for about 10% of all LDLR mutations. Here is a map of the major LDL receptor domains. And next, I will show you what parts of the gene the structural variants that we found in this cohort affect. So by applying the structural variants workflow provided by the Genomics England, we found a deletion of promoter, exon 1, and part of intron 1. We also found a deletion of exon 5 and its surrounding introns, a deletion of exons 13 and 14, plus their surrounding regions, and the largest, over 5 kb, deletion of exon 15. For the first time, we were able to give the exact size of the deletion and the precise breakpoints at which the mutations happen. So this part of analysis explained FH in further 4 probants and 5 relatives. So next I will pass on to Martin, who will show you his efforts in identifying mutations in untranslated regions of the gene, and I will be back.
So my research has specifically focused on a particular structure within the 5' UTR called upstream open reading frames. Now, an upstream open reading frame is defined by an upstream start codon within the 5' UTR, which results in a reading frame upstream of the coding sequence. And these upstream start codons can attract scanning pre-initiation complexes and can be translated by a ribosome. And if this occurs, there is a lower likelihood of the downstream coding sequence to be translated. So functionally, these upstream open reading frames play a role in regulating mRNA translation of the exon coding sequence. Translated upstream open reading frames can reduce expressed protein levels by 30 to 80% across the genes they are present in, and exist in about half of all genes, suggesting they play an important role in the regulation of gene translation and therefore gene expression. And additionally, some genes can contain multiple upstream open reading frames where they can have a cumulative repressive effects on translation. So what makes these upstream open reading frames interesting is that there can be variants which alter the amount of upstream open reading frames in a 5' UTR or the structure of existing upstream open reading frames. And this has consequences for downstream translation. So a variant which introduces an upstream start codon can create a new upstream open reading frame, decreasing downstream translation. Conversely, a variant which knocks out an existing upstream start codon removes the existing upstream open reading frame, meaning there's an increased chance of translation starting on a coding sequence, which increases translation effectively. An upstream stop loss variant can extend an existing upstream open reading frame into the coding sequence. And this creates an overlapping open reading frame, which if translated completely blocks out translation of the coding sequence. If the effect of decreased or increased translation is enough to cause damaging phenotype, then these variants are mechanically causing haploinsufficiency or toxic gain of function respectively. And so it's been observed for the last couple of decades or so that these variants can alter the 5' UTR to have more or less upstream open reading frames. They can be pathogenic in genes where haploinsufficiency or toxic gain of function is a known mechanism of disease. Additionally, a factor which determines the effect of an upstream open reading frame altering variant is the Kozak consensus sequence. And the Kozak consensus refers to a particular pattern of nucleotides surrounding a start codon that influences the attraction of translational factors and the pre-initiation complex, where the -3 and +4 bases surrounding the start codon are the most conserved. So this chart on the right here from a 2021 study from Wright and colleagues shows a luciferase assay comparing expression of MEF2C when two different upstream AUG gain variants are introduced. And as you can see, the stronger the Kozak sequence strength, the more the upstream open reading frame which gets translated, leading to decreased protein expression. So for the last couple of years, detecting these upstream open reading frame altering variants has been quite rare until Zhang and colleagues in 2020 developed the UTR annotator plugin for the popular VEP software, which is often used to annotate genetic data. And so I performed annotation using this tool of the 550 and 544 FH participants' whole genome sequencing data, focusing on the FH genes where haploinsufficiency or toxic gain of function was a known mechanism of disease. And I found a single variant substitution of C to G 35 bases upstream of the canonical first exon of LDLR, which had a moderate Kozak strength. And so this upstream start codon, if translated, then results in an overlapping reopen reading frame, which is frame shift to the canonical reading frame, which covers the entirety of the first exon of LDLR, which essentially skips translation of the first exon. And this patient also had no tier 1 or 2 variants that explain their phenotype. So I would then like to follow this up with a luciferase assay to then confirm if there is in fact a decrease in LDLR protein expression. And it would also be interesting to see if this variant has a similar functional effect to exon 1 deletions. I think this is quite exciting because it shows how whole genome sequencing can capture these regions that maybe at one point we don't know how to effectively annotate, but maybe in a few years we can always come back and learn more about them.
Thank you, Martin. Yes, so it's an exciting part. So we think we have at least one proband with that variant, and we are currently in the process of requesting collaboration through Genomics England to follow up, hopefully, this family. So let's have a look at other genes that are known to cause FH. ApoB is one of the largest genes, but remains highly understudied, and this is probably because of its size. So many historical FH genetic tests would focus only on the region containing the most common FH mutation in the exon 26. We also checked for this mutation in our cohort and found actually 6 probants having that variant. Another known FH mutation was found in the same codon, a variant that is more prevalent in individuals of Asian background. So we also identify 1 proband with that variations. We next extracted ultra rare and variants that were unique to the FH cohort in comparison to the rest of 100,000 genomes. And this analysis found 7 variants, 5 of which are considered to be novel, as they were not present in GNOMAD browser either. These are great candidates for further investigation, and I'm hoping to develop a mass spec-based assay to further study these variants. There is also a variant that isn't unique to the FH cohort, but has come up as enriched in FH in a single variant association testing. And the variant, again, requires further investigation. Let's go back to the ApoE that I mentioned before. ApoE encodes a component of several classes of lipoprotein particles, and it is known to interact with the LDL receptor. I mentioned that relatively recently a mutation, an in-frame deletion of a single amino acid has been shown to segregate with FH in a large family. And 3D protein modeling suggested that it is a gain of function mutation that binds strongly to the LDL receptor and blocks its recycling. As this is a relatively recent finding, most of the previous genetic testings for FH would not have included this variant. Therefore, we were expecting to find some carriers in this cohort. And indeed, we found 10 probands carrying the variant. There are also 5 other non-FH participants in the 100,000 genomes who have the variant, and we have been collecting data on their lipid profile. I must say the response from the referring clinicians was rather poor, but hopefully we will be able to show either that there is an association of that variant or there isn't. It is still unsure if that is true FH causing variation. So just to summarize so far, our initial findings provide FH diagnosis to 15% of the cohort participants, with majority of them being due to LDLR mutations. 4% of variants in known FH genes are VUS variants of unknown significance, and they are great candidates for future follow-up. And of course, question keeps bothering me why those variants were not identified by standard methods. This is something that I hope to follow up by contacting the referring clinicians and see if those individuals really had the original DNA test for FH. I thought perhaps there were some deadlines in sample submission, so maybe there was a specific center that would be submitting patients that haven't had initial DNA test. I don't want to be naming or shaming anyone here, but when I plot the GMCs, referring GMCs for those known mutations, there doesn't seem to be any pattern, so they come from many different centers. So I'm not quite sure what the issue was. But of course, the elephant in the room here is probably this orange part of the pie chart. What causes high cholesterol in the majority of the FH cohort in 100,000 genomes? Perhaps one of the answers is a polygenic component of FH. And this would be due to the burden of common LDL cholesterol rising alleles. Back in 2013, we designed a 12 SNP score to assess polygenic component in familial hypercholesterolemia. So this was based on genotypes from 12 SNPs, which had the biggest effect, beta effect on LDL cholesterol, from the most powerful then GWAS study. And this work has been conducted by Dr. Antoine Wimbert, who have calculated this 12 SNP score using the whole genome sequencing data in the whole cohort of 100,000 genomes. He also did it in the FH mutation negative cohort, and what the finding was that the 12 SNP score was significantly higher in the mutation negative individuals. But of course, there is a significant overlap between these two. But overall, if you take individuals with 12 SNP score over 75th percentile, 29% of the mutation negative cohort has such high score. Obviously, last week we had news of new data release in the 100,000 genomes provided by the Genomics PLC, the PRS for coronary heart disease and coronary artery disease. So what we're doing now is we want to repeat this analysis using these much larger polygenic risk scores that capture not only LDL cholesterol, but other aspects of the coronary heart disease, and see if that difference separation here is much clearer.
Sorry, I have to wrap up now.
Yes, okay, so my summary slide is that it is important to identify FH mutations that really enables early diagnosis and cascade testing of the relatives. Using whole genome sequencing, we were able to provide diagnosis for 15% of the patients, and these are due to mutations in known genes. Whole genome sequencing is effective at identifying structural variations. And also we identify 4 rare non-coding variants, and Martin covered one of them. There are a few other ones, deep intronic variants, that are unique to the FH cohort. Novel missense variants in ApoB require further investigation, and the polygenic component is present in 29% of the sequence patients. We are currently conducting gene burden tests for any other gene in the genome to see if there are any novel causes of FH underlying the phenotype. The biggest limitation at the moment we have is the lack of lipid measurement data for not only for the FH cohort, imagine, but also for the rest of the 100,000 genomes. So that only limits us to the case control comparison. I just want to also mention that FH is a medically actionable disease and is part of the additional finding analysis. So there's ongoing work on that as well in the rest of the 100,000 genomes participants. Last but not least, I would like to acknowledge my very important collaborators, work led by Steve Humphreys, Dr. Anton Bird, Maggie Williams from Bristol, collaborators in Portugal who we're working together on the ApoB variants, and of course, Genomics England, especially the support team in bioinformatics. Thank you very much for your attention.
Thank you, Martha. So we have pretty much run out of time. There are some questions in the Q&A. I think you're going to have to answer most of them offline. I'll just select one of them. There's a comment. Is there evidence that receptor negative mutations result in a more severe phenotype than receptor defective mutations?
Yes, valid question. Yes, so recently we've looked at a cohort of children with FH, and that was the biggest study of children with mutations, over 3,000 individuals. And we've made, what we noticed is that those with loss of function mutations had significantly higher pre-treatment cholesterol levels in comparison to those with sort of missense variants. So there seemed to be some association. And
the interpretation of family data may be complicated by the overlay of environmental factors that influence lipid levels. So, you know, is that where you're saying actually you need lipid information measurements to help with diagnosis?
Yes, so that is true. So LDL cholesterol, as we know, is influenced by dietary factors, lifestyle factors, also by the polygenes. So we need to get the bigger picture. Ideally have rare variants, have also polygenic risk score, and that way we can start, and then we'll see how if there are major differences in terms of families. Most of the variants usually they segregate clearly in families, but yes, it seems like the relatives, especially children, have much lower cholesterol levels than the parents who are also affected. So it is complicated. It's definitely not very simple, but it's a good start, I think, for interpretation of the variants.
Okay, so we're out of time. Thank you very much, Martha, and thank you, John. Before you two, I think you can possibly answer some of those questions in the Q&A before everything closes. Maybe they can be recorded in the distribution that comes afterwards if there's anything to comment on. Just to say, the next seminar in this series is in June. I think if Zan's got the slide, we can put it up. It's probably in the mails you've received already. Just to remind you of that. So if you're signed on, then you'll get mails about all this series in future. Okay, so thanks very much to the speakers. Thanks for everybody attending, and look forward to seeing you all in the future.